Namaskar. I welcome all of you for this online CTET training program. And in this session, we are going to learn about the microorganisms. The next chapter, microorganisms. And I am K. Madhu, a senior research fellow from Regional Institute of Education, Mysore. So this is from paper 2 of science. This chapter is about microorganisms, and this chapter we divided it into two parts, that is part 1 and part 2. So in part 1, in this session, we are going to talk about the classification and useful microorganisms. The next part, that is part 2, we will be focusing more on the harmful microorganisms and how they are harmful and what kind of diseases they are going to cause to all the human beings. So this is what it is. And so how we are going forward with this session is, so first we'll do, as usual, how we do in our normal sessions, like we do trend analysis of the concepts, the most repeated concepts which were tested. Then we'll discuss the concepts that are most relevant. Then we'll try to solve PYQs, that is previous year questions based on the concepts that we're going to discuss. And these are the concepts which were repeatedly being tested in the CTET. At the end, we'll have a quick recap. So what CTT asks about useful microorganisms? So with respect to useful microorganisms, so mostly it is about the uses of microorganisms. Again, in the uses of microorganisms, especially the use of yeast, okay, then and medicinal uses, especially the antibiotics, how antibiotics are useful and to all the human beings with respect to their health, and nitrogen cycle. So they are the major areas where CTT focuses with respect to microorganisms. And also apart from that, the scientist discovery matching. This is one more most important concept, like where we have to, most of the questions will be from matching the scientists with their discoveries, and also matching the microorganisms and their uses. So matching organisms, especially yeast, how yeast is useful, how lactobacillus is useful and all. Then nitrogen cycle is also important, but mostly it is the matching of scientists with the discoveries and the matching of microorganisms with their uses in our daily life. So first let us classify and understand the different microorganisms that are present around us. So there are totally 5 types, okay. These are the different microorganisms and how they are different, okay, how they are different and what is their role. Let us understand. So the first and foremost type is bacteria, okay. Bacteria is prokaryotic in nature. So we need to understand the structural differences between the microorganisms to understand different types. The first one is bacteria. It is a prokaryote. We discussed about it when we were talking about the cell, okay. When we were discussing about cell, we also talked about bacteria. It's a prokaryotic organism. The other organisms are eukaryotic in nature, okay. Eukaryotes we can see here. So the bacteria, fungi, algae, protozoa, viruses, these are the major classified major types of microorganisms that are there. So bacteria is it is unicellular in nature and it is prokaryote. It doesn't have a nucleus. Prokaryote cells or prokaryotic organisms won't have the nucleus, proper nucleus, and definite organelles won't be there, okay. Whereas eukaryotes will have well organized cells. They have proper nucleus, nuclear membrane, and other organelles also, which will be seen in fungi, algae, protozoa and viruses. So they don't have nucleus, they are single celled, so most abundant life on earth. So they are everywhere, omnipresent they are. What about fungi? It is eukaryote, okay. So it has chitin wall. It is made up of, it has cell walls which are made up of the material chitin, okay. So it doesn't have chlorophyll, so it doesn't perform photosynthesis. So the best example which is being uh, repeatedly tested in CTET is yeast. So remember yeast, it is the unicellular fungi, okay. It is unicellular eukaryotic fungi which will be frequently asked in CTET. Then we have algae, which is eukaryote, okay. So it has chlorophyll, so it does photosynthesis, so autotrophic in nature, makes its own food. Then Protozoa, which is also an eukaryote, unicellular. Then this is the difference between bacteria, okay, versus protozoa. So whereas bacteria is unicellular prokaryotic, whereas protozoa is unicellular eukaryotic. That is a difference between the cellular organization between these two. Even fungi, algae are also same. Okay. So yes, so it is animal like because it undergoes heterotrophic nutrition. Amoeba, paramecium are the examples. Amoeba, we have discussed about how amoeba does nutrition, how it extends um, its pseudopodia for capturing a food item and also for the movement purpose. So amoeba is one example, another is paramecium, two examples of protozoa which are eukaryotic organisms and single-celled in nature. Whereas we are calling protozoans as heterotrophic and they are animal-like. Why not fungi? Why are we not calling fungi as animal? Because it doesn't, that one thing is it doesn't fall under plants because it doesn't have any chlorophyll. Other is it doesn't fall on animal also because it doesn't it doesn't have same nutrition as animals as animals follow. It has a different kinds of nutrition. It is saprophytic in nature. Saprophytic in nature means they completely depend on the dead or decaying matter. So whatever the dead organisms will be there, they will be dependent on these dead organisms for their nutritional purposes. How these fungi will go and they release digestive juices. They go sit on the dead and decaying matter. They release the digestive juices. That's how they digest the food material and take it. That is their nutrition. So that is why it is completely kept in a different category. That is fungi. So it is not kept in animal kingdom. It's not in animals. That is because of it has a saprophytic nutrition. So saprophytic nutrition is also one type of heterotrophic nutrition, but then how animals depend on other organisms and how fungi depends on other organisms, there is a lot of difference. So animals depend on plants and other animals which are living and which will be dependent on the living organisms. Okay, whereas fungi completely depends on the dead and decaying matter, dead and decaying organisms. Okay, so that is the main difference between fungi and protozoa and why they are kept in two different categories, though they are they follow heterotrophic nutrition. Then at last we have viruses, very peculiar microorganisms they are because they don't have any cell structure. They are not cells, to be frank. They don't have kind of a nucleus or cell organelles or cytoplasm or cell wall, nothing. Okay, they are just a nuclear material we can consider. It has either DNA or RNA. Okay, it's not truly living. Okay, it can only reproduce inside a host cell. Host cell means whenever it enters an organism, then only it becomes active. Then only it can reproduce. If it is outside, it cannot reproduce at all and it won't show any sign of the living organism. No reproduction, no nutrition. It is just still like a non-living thing. That is why it is very peculiar microorganism. And it doesn't have any cell wall or any cell organelles and all. Just and it's a nuclear material, okay, which is enclosed in a protein coat. So these are the 5 types of microorganisms: bacteria, fungi, algae, protozoa, and viruses. So let us move forward and understand how these are useful. Especially in this session, we are going to focus more on how these microorganisms will be useful for us in our life, and we'll be solving questions and we'll be focusing on questions based on the useful microorganisms. So microbes in food, okay. So in food industry, how microorganisms help us in food preparations, especially curd preparation, bread preparation, and alcohol preparation. So commercially, microbes are being used in our daily life, to produce different kinds of foods. So examples are already given here: curd, bread. In baking industry they are used. Even the dairy industry also they are used, and they are used in alcohol industry also. So what are these microorganisms which are being continuously tested and asked with respect to their uses in CTET? So there are 3 most important microbes here. So lactobacillus, yeast, and again yeast. How these are the majorly used microorganisms for the food preparation. So lactobacillus helps for setting of curd. Okay, whereas yeast, it helps in bread preparation and also production of alcohol and wine. Okay, lactobacillus, so it sets curd. So this will convert the milk, lactose in the milk. So the starch inside the milk is called as lactose. So this starch, this lactose will be converted into lactic acid. Okay, and this lactic acid, helps the milk proteins to solidify and coagulate and make curd out of it. Okay, this is how lactobacillus, when it is introduced into the milk, okay, boiled milk, uh, at certain temperature, so it starts reproducing. So when it starts reproducing, it will convert lactose into, it will act on the lactose, break the lactose and make it to lactic acid, and that will later help for preparation or setting of curd. Then yeast helps in baking industry. Okay, it helps in preparation of bread which are fluffy in nature. So when the yeast starts fermenting, we discussed in previous sessions also that yeast help in fermentation. So these will um, go and do fermentation on sugars present in the different food materials. So this fermentation of sugar will form alcohol plus CO2. Okay, CO2 gas will be released and alcohol is also produced. So this CO2 bubbles expand in dough. So whichever dough it is, so they will expand and in the dough, and bread becomes more spongy in nature. The cakes, the bread that we see, it is the fluffy nature of the the cakes and breads is because of the expansion or the bubbles that were created by CO2 production during the fermentation by yeast. Okay, so this is one more example. Then yeast also helps in alcohol. We saw here alcohol plus CO2 is released, but whereas what happens to alcohol here in baking industry while baking, while preparation of cakes and breads, so this alcohol gets evaporated completely. So this alcohol gets evaporated completely. Only the dough and the expansion of CO2. Okay, the dough, in the dough, the CO2 is released. CO2 occupies the spaces in between the dough and it becomes pulpy in nature, whereas alcohol is completely escaped and evaporated. So we get only the bread and the cakes that we talk about. Whereas in especially alcohol preparation, this alcohol is taken, alcohol is it is make sure that alcohol will be stored. Okay, same yeast fermentation, ethanol is kept. Okay, it is made sure that alcohol won't gets evaporated, it is kept. Okay, then not CO2, focus is here on ethanol. So this fermentation that we talked about, in yeast, whether to prepare bread or alcohol, it happens with in the absence of oxygen, anaerobic respiration. So we do talked about in physiology, when we talked about respiration, we talked about two types of respiration, that is, one is aerobic respiration, another is anaerobic respiration. So fermentation comes under anaerobic respiration. So this yeast, okay, the fermentation was first discovered by Louis Pasteur in 1857. We'll also talk about the different scientists and their discoveries with respect to microorganisms. Yeah. And how microorganisms are helpful in health, okay, for healthy purposes. So there are different medicines that are prepared using microbes which we are daily using. So let us see a few of those medicines which are prepared from microorganisms and which are very important from the perspective of CTET examination. So penicillin, it was the first antibiotic. So remember this fact, penicillin is the first antibiotic that was prepared. It was discovered by Alexander the Fleming in 1929. Okay, it was from the mold penicillin, Penicillium notatum mold. Okay, then antibiotics are also prepared which can kill the bacteria only. Antibiotics kill only bacteria but not the viruses. Why not viruses? Why only antibiotics work on bacteria? Because we discussed in the first slide that viruses are not truly living. They are non-living outside, but when they enter inside the body, then only they show living signs. And why antibiotics doesn't act on viruses? The main reason behind it, as we discussed, the viruses doesn't have cell organelles. It doesn't have a cell membrane, it doesn't have proper cytoplasm, it doesn't have ribosomes and other organelles on which antibiotics will be binding and acting. So why antibiotics will act on bacteria? Because bacteria is a cell. Though it is a prokaryotic cell, it is a complete cell, okay? It is a cell, right? It has a cell membrane, it has cell wall, it has all the other organelles, it has DNA, nuclear material and all. So it has, the antibiotics act on this cell, the different parts of cells. Whereas virus is not a cell, so antibiotics doesn't act on the virus. Virus have no cell wall, so no metabolism, so antibiotics cannot do anything with respect to viruses. Then Edward Jenner, the scientist Edward Jenner is the one who came up with, who discovered vaccine for smallpox in 1798. Okay, so he is called as father of immunology. We'll later discuss about the scientists and their discoveries. Then vaccine mechanism. So we have antibiotic. We have antibodies that are prepared from microorganisms. Then we have vaccines. So what are vaccines? Vaccines are dead or weakened pathogen. So vaccine is given to individuals which will consist of the dead and weakened pathogen of particular microorganism, okay, particular disease. So that will initiate the immune response. So an immune response will be generated in the individual by introducing a dead or weakened pathogen into them. So antibodies will be automatically, naturally produced by the human body, and that's how immune response is created, and people become immune to that particular microorganism in the future encounters. So these are the different uses in the medical field, in the health sector, from the microorganisms. So nitrogen cycle, so there are majorly 5 steps, but the major focus in CTET is on the scientists and the discoveries and the microorganisms and their uses. But this is also one of, little importance is there from CTET perspective. So let us just quickly understand nitrogen cycle, its steps basically. So what are its steps and what is the sequence between the steps? So this was important. So from top to bottom, the sequence is in this order. So 1, 2, 3, 4, 5, it is a cycle which happens. So first what happens, nitrogen fixation will happen, then assimilation will happen, then ammonification, nitrification and denitrification. So what happens in each step is in fixation, nitrogen will be fixed by the rhizobium, okay, rhizobium bacteria, okay, they are present in root nodules. So nitrogen from atmosphere, that is N2, will be fixed in the soil. Then what happens, assimilation. So plants absorb this nitrogen from the soil, that is assimilation process. And plants, why do plants absorb, why do plants require nitrogen? Because they require nitrogen for synthesis of their proteins. For plant proteins they required. Then ammonification. So when these plants or the animals which ate these plants die and they decompose, so again ammonification takes place with the help of saprophytic organisms, where again from the dead bodies the nitrogen enters into the soil. Then nitrification happens, okay. So plants, so again it is they like in the soil, again it is available once the nitrate enters again soil, again it is available for plants and plants for the for absorption from the plants. So nitrification, then denitrification, okay. The pseudomonas converts nitrates into N2 gas again. So from again soil it will be released into, it will be converted into N2 gas which will enter into the atmosphere. So it's a cyclic process, okay. So all these processes are happening with the because of the the microorganisms. Rhizobium is the one which fixes atmospheric nitrogen again to the from soil to atmosphere, it is pseudomonas that is doing. So this is about nitrogen cycle. which happens through with the help of microorganisms. So let us understand and match. So matching is one type of question that is frequently asked with respect to microorganisms, whether it is useful or harmful microorganisms. So let us understand the scientists who and their discoveries and the years also. Okay, the timing is also important. So it is Leeuwenhoek in 1670. Okay, what did he do? So he observed the first living microorganism. Okay, he is the first one to observe the first living microorganism. Okay, he is called as father of microbiology. So from him, actually, it started the observation of living organisms. Okay, the first microscopic living organism. That's why he is called as father of microbiology. Then Louis Pasteur in 1857. So it was fermentation, he explained, and also developed pasteurization, uh, where boiling of milk and the setting of curd, all that he spoke. Okay, so he also disproved theory of spontaneous generation. So mostly we'll remember Pasteur, Louis Pasteur, okay, for his pasteurization technique. Then Edward Jenner in 1796, he is called as father of immunology. Okay, so please remember all these facts. So he is the first one to prepare first, he developed the first vaccine, that is for smallpox. Then Alexander Fleming in 1929, okay, treatment for bacterial infections through penicillium. Then Robert Koch 1876 to 82. So he discovered uh, anthrax and TB and bacteria. Okay, so he is called as father of microbiology. Just remember what are the names that are given to the scientists and what they have contributed. So these please remember. And yes, there are different relationships that will be there in our with respect to micro the micro level world. Okay, so symbiosis, parasitism, and decomposes. So we are talking with respect to how microorganisms exist, how they exist with respect to the other organisms, and how they are helpful to other organisms we saw in for human beings, and also how they are helpful for other organisms apart from humans. So symbiosis is a relationship where both organisms benefit from each other. Okay, it is rhizobium and in leguminous plant. Okay, rhizobium is will be present in the root nodules of leguminous plant. So they help each other. Okay, so rhizobium fixes the nitrogen for the leguminous plant, and the leguminous plant provide food to the bacterium. Then parasitism. This is one way how certain microbes depend on other organisms. Here the Cuscuta is given, which is a plant again. So let us not see parasitism here, because we'll be talking about harmful microorganisms later in the next part. So let us not talk about parasitism here. Okay, we'll talk about in the next part. Decomposes, they are also beneficial being decomposers. Microorganisms are beneficial to us being decomposers. How? Because they clean the environment by cleaning all the dead and decaying organisms. So they are saprophytic. There are saprophytic bacteria and fungi. So fungi is mostly saprophytic. There are bacteria also which does the decomposition of dead and decaying matter. So this is how different microorganisms are helpful uh, in the world, and this is how humans are getting benefited, and all other organisms also. So these are few facts that we discussed, and few concepts which we discussed where focus was there from CTET. So let us see how questions are being asked, and let us try to solve few questions based on these concepts. So as I said, it is mostly matching type. In this chapter, the questions are mostly of matching type, giving organism, microorganism, and matching it with the uses, giving scientist names and matching with the discoveries, or giving discovery and asking for matching with the scientists. So here let us see the question. So here there are two sides, two different things are given. On one side, the microorganisms are given A, B, C, D. Another side, the roles are given. The roles of microorganisms. Okay, what is their role in the different things? So we have to match the microorganisms with the roles. So A is lactobacillus, B is protozoan, C is rhizobium, D is virus. So we have to match them. The roles given are like this: nitrogen fixation, then AIDS, malaria, then curd. So lactobacillus is related to what? Is it related to nitrogen fixation or AIDS, malaria or curd? So we discussed that lactobacillus is the bacterium which converts milk into curd. So lactobacillus and curd are related. Then protozoa. Protozoa causes malaria. So this is related to the harmful effects or harmful nature of microorganisms. And C is rhizobium. So what is the role of rhizobium? It fixes the nitrogen. Then virus causes AIDS. So AIDS is a disease which is caused by virus. So the correct answer is this. Okay, lactobacillus is curd, protozoa is malaria, okay, rhizobium nitrogen fixation, virus is AIDS. So this is what it is. And let us move to the next question. Okay, it's from 2022. And so the question is, A breaks down sugar into alcohol. Okay, A breaks down sugar into alcohol, and C in the absence of oxygen. C forms bubbles in dough, increasing its E. Identify ACE. So it is a big statement where we have to identify the different we have to identify what is A, what is C, and what is E in the statement. So let us break down the statement now. So A breaks down sugar into alcohol. Okay, let us see till here. A breaks down sugar into alcohol. So which microorganism breaks sugar into alcohol? So we talked about fermentation concept. In the fermentation concept, yeast will act on sugars and they convert into alcohol. Okay, and CO2 is released. So the first sub statement, that is A breaks down sugar into alcohol. So it is definitely is. So we have to look for the options which has yeast. So first one doesn't have yeast, so definitely wrong. Second one also doesn't have yeast, so second one's wrong. So only fourth and third have yeast. Okay, you can see here. So next we'll move forward. C forms bubbles in the dough. So C forms bubbles in the dough. So we discussed how bubbles and the fluffy nature of bread will be coming. So from where it will be coming? It is because of the carbon dioxide release, carbon dioxide expansion. So both have carbon dioxide. So both are there. So let us go for E. Let us try to identify E. And so C forms bubbles in dough, increasing its E. Identify AC. So when C forms bubbles in dough, it increases its E. That means the C increases E in dough. So what does C increases? C is what? Carbon dioxide. So what does carbon dioxide increases in the dough? It increases its volume. Okay, it's not aerobic. Aerobic is a type of respiration. So the third one is the right answer. So whenever you see these kind of big statements and asking you to identify the components given, so please try to break the question first. Try to understand by breaking the question. Then you'll be able to understand the question easily, and you'll be able to relate the question to the options more appropriately. So let us move forward to next question. Yeah. So yeah, A is yeast, C is CO2, E is volume. Next, again one more matching question here. Okay, scientists and their discoveries. So we discussed in one particular slide about the scientists, what they are called and what are their discoveries are. So here different scientists are given, and their contributions and their contributions or the microorganism they worked with are given. So A is Fleming, Jenner, Louis Pasteur, Koch are given. Then anthrax bacterium. fermentation, penicillin, and smallpox. So it is just matching the scientists with uh, their contributions or the microorganisms they worked with. So most frequently asked questions from microorganisms are matching the scientists and their discoveries, apart from the matching of microorganisms with their uses. Then in which process bacteria in which process is bacteria not used? So they are given 4 different processes where bacteria is not used. That means the use of bacteria. Again, we have to match, not matching exactly, but we should identify where bacteria will be used, where fungi is used, in which process and all. So is it nitrogen fixation? So nitrogen fixation, rhizobium bacteria definitely is used. Then formation of curd, we know that lactobacillus is used. Then baking of bread, which one is it, bacteria or yeast? It is yeast. Okay, fermentation of sugarcane. So even fermentation is done by few of the bacteria also, especially in sugarcane. So it is baking of bread. We especially discussed in baking process, the fermentation of sugars in the dough takes place, and so it is yeast that does. Okay, it's not bacteria. So it is baking of food. Whereas nitrogen, curd, and bread, yeast, everything is right. So one more question from 2013. Okay. So two organisms live together. So this question is about the relationship between the microorganism and the other organism. So two organisms live together. One provides shelter and nutrients. The other prepares and provides food. This association is called as. So both are benefited here. So always remember, if both the organisms in a relationship are getting benefited, it is symbiosis. Okay. Heterotrophy is a nutrition. Parasitism is also one more relationship where only one gets benefited, the other gets harm. Okay. Autotrophy is also one type of nutrition. So these are a few questions that tell us how questions are being asked, mostly matching with respect to useful microorganisms also, or even harmful microorganisms also. So matching type of questions are more and repeatedly asked. So the symbiosis is the right answer. So few facts that we should remember from this chapter or from this topic are, so yeast is the unicellular fungus, and curd is related to lactobacillus bacteria because that's what helps for setting of curd. Jenner is related to smallpox. Then Fleming, it's penicillin. Okay, scientists and their contributions. Rhizobium, symbiosis. Then scientific match. Okay, all. So these are the few areas where we need to focus more in this topic. Um, so these facts also, please, you may remember, because whatever the facts that we are discussing at the end of the session are the most repeatedly tested facts they are. Okay. So with this, we'll conclude this session related to microorganisms classification and their uses. So let us continue in the next session where we'll be discussing about the harmful microorganisms and how they are harmful for the human beings and also plants and animals. So let us conclude now. Thank you so much.